Again, like we mentioned in terms of metabolism of the myocardium, it can only operate aerobically. So it absolutely needs oxygen in order for it to do this. It also has 3 times higher oxidative capacity in comparison to skeletal muscle. So there's a high amount of mitochondria, which is the organelle of the cell that is responsible for aerobic metabolism. And again, we'll talk about that in more detail coming up maybe later this week. And it's also really good at long chain fatty acid metabolism. And again, the nice thing about this, omegas, they say like, healthy fats for your heart. You've heard that before basically, but we can use those for a fuel source basically for the heart. And that's what they're talking about here. So we can use fat as our fuel source within the myocardial tissue. Okay. Second part of this lecture, basically, in real life, this is where we would have taken a break. So if you need a break, please, by all means, pause this and take a break. Okay, so now we're going to move forward and talk about the cardiac conduction system. And the cardiac conduction system is basically the electrical system which allows for us to stimulate the heart contraction. So we've got this electrical system again that will basically allow us, like I said, to stimulate the heart. So the system is made up of several components. We have an intrinsic natural rate. So if we didn't have the cardiac conduction system, our heart would beat at an intrinsic rate of 100 beats per minute. Okay. Our SA node paces us at about 60 to 75 beats per minute. The SA node is known as the pacemaker, and he's located on the posterior wall of the right atria. And the electrical impulse is created here, establishes what we call a sinus rhythm, and this spreads the impulse to both of the atria, so both the left and the right atria, and then it sends it down to the AV node. The AV node, or the atrioventricular node, has its own intrinsic rate of 40 beats per minute. All right. So once the signal reaches the AV node, it pauses here for 0.13 seconds. This allows for the atria to finish their contraction completely. So again, this pause is what allows us to hear like the lub-dub, 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 if you were to hear a heartbeat. So the atria are going to completely empty into the ventricles, and this allows us to maximize ventricular filling. And this is actually known as the atrial kick. So when we say the atria contract, I know like what they probably taught you in high school was that all the blood's in the atria, and then the atria contracts, and all the blood moves to the ventricles, and then the ventricles contract. Well, that's not actually true. So like 80 to 90% of the blood goes directly into the ventricles. And the atria is a little bit of a baby and doesn't really actually do that much work. So the atria, when it contracts, it only moves 10 to 20% of the blood on down to the ventricle. So the ventricle already is like pretty full. And then once the atria contracts, this accounts for what we call the atrial kick. And again, that accounts for about 10 to 20% of blood filling. Okay, so the atrial kick occurs. The AV bundle, also known as the bundle of His, bundle branches. So we've got a right and a left side, and then Purkinje fibers. The spread of the signal is going to move very rapidly from the AV node down to the bundle of His, to the bundle branches, and then to the Purkinje fibers. Right. So let's talk about where each of these are located, and then we'll show you a picture of these. So the AV bundle, or the bundle of His, travels along the ventricular septum, or kind of like the border, or the like the border between the two ventricles, basically. The amount of muscle mass that separates the two sides, the right and left side. And it connects the right and left bundle branches towards the apex of the heart, which is the bottom of the heart. And then the Purkinje fibers are the terminal branches, and they're going to send their impulse 6 times faster than any other part of the conduction system. So rapid conduction allows the ventricles to contract at virtually the same time, and this causes the ventricles to contract from the apex up. So again, like I said, the spread of this signal is very quick once we reach that bottom half of things. Okay, so here you can see all of these components of the cardiac conduction system. Also, two other things I want to point out. The apex is the bottom of the heart, and the base is actually the top of the heart, which is kind of like counterintuitive to what I would think. So it's like opposite of what I think it should be, but nobody put me in charge. So anyway, so the base is at the top, the apex is at the bottom. All right, so let's again review the components of the conduction system. So you've got that sinoatrial node, which is located on that posterior side of the right atria. That serves as our pacemaker and sets the rate of the cardiac conduction system. So he sends a signal to the atrioventricular node where we get a little pause for 0.13 seconds. That allows the atria, both right and left side, to contract fully, and that causes our atrial kick. Once we have the atrial kick completed, the AV node will quickly send his message to the bundle of His, the left and right bundle branches, and then finally onto the Purkinje fibers. Again, the spread is very rapid at this point, and that will allow the ventricles to contract in unison, and they're going to actually contract from the bottom up, which makes sense if you think about where their vessels are. So there's no vessels coming out of the bottom of the heart. So it's important that we contract from the bottom up because the vessels that blood's going to leave out of are kind of like at the top of the heart. So that's important for us in terms of functionality. Okay, so an EKG or an electrocardiogram is probably something that you've heard of before, but basically this measures the electrical activity of the heart and can be recorded to monitor cardiac changes or diagnose potential cardiac problems. And again, we'll actually get the opportunity to do a EKG on everyone in the lab. And basically, if we were to do a full EKG, you're going to place 10 leads all over different locations of the body, and they're going to detect electrical conduction as it flows through the heart. So basically, the reason that we have 10 different locations is it allows us to kind of take a snapshot of the heart, or take a video, more accurately, take a video of the heart from 10 different viewpoints. So it basically allows us to more fully kind of understand what's going on in terms of the electrical signaling within the heart. Okay, so this shows you what an EKG printout would look like. So the different waves that we have, we've got the P wave, PR, QRS interval, ST segment, ST interval, and QT interval. So an interval, like if you say ST interval, it contains the T itself, so it contains the waves mentioned. If you talk about a segment, you're not including the actual wave. So a segment looks at like the time between, or basically like the time between each of those components that you're tagging. So like the ST segment doesn't include a wave, and the PR segment doesn't include a wave. Let's see, what else would we like to define here? So the P wave represents atrial contraction, or atrial stimulation, you could say. The PR interval would tell us the amount of time it takes from the stimulation of the atria to the stimulation of the ventricle. The QRS interval, you can see here, has the highest amplitude, so that tells us it requires the greatest amount of strength, and that's the contraction of the ventricles, which makes sense because we know that they need to have a stronger contraction in relation to the atria. The ST segment tells us basically the lag time between the contraction of the ventricles and the recovery of the ventricles. Okay, so the T wave represents ventricular repolarization, or allowing the ventricles to return to their resting state. Okay, and the RR interval is what we can utilize to determine heart rate, basically. So we can calculate how far it is from R to R, so how far it is for one cardiac cycle, and use that to determine how many times the heart is contracting per minute. Okay, so we just covered all of these, so hopefully you wrote those down from the previous slide, but I'll briefly say them again. So the P wave tells us atrial depolarization. The PR segment, again, tells us the relationship between the stimulation of the atria and the stimulation of the ventricles. QRS complex is the ventricular depolarization. T wave is the recovery or repolarization of the ventricles, and the RR interval is one complete cardiac cycle. And again, we can utilize the RR interval to determine what the heart rate is. Okay. So in terms of extrinsic control of the heart, although we have some intrinsic control or factors that we can control within the heart, it's generally under the control of extrinsic factors, or other people that don't live there are telling them what to do from afar. And you'll never guess who's telling the heart what to do. Yes, I'm sure you did guess. It is the brain who is telling the heart what to do. And there are several different factors: parasympathetic nervous system, sympathetic nervous system. The endocrine system can also play a role in determining the contraction level, and not only the frequency, but also the strength of contraction within the heart. So the parasympathetic nervous system is going to allow us to slow down our heart rate. Okay, so I mentioned before we have an intrinsic heart rate of 100 beats per minute. But most of you know a normal resting heart rate might be somewhere between 60 to 70, somewhere in that range is reasonable. And so this tells us that while at rest, the parasympathetic nervous system has control over our hearts. And basically what happens is the vagus nerve is going to send an impulse to the SA node. And at the SA node, it's going to release acetylcholine. And we've talked about acetylcholine before. And once acetylcholine is released to the SA node, this decreases the firing rate and therefore decreases our heart rate. Like I said, at rest, we have a dominant vagal tone, which again tells us that the vagus nerve is the one influencing this, and the vagus nerve is getting his information from the parasympathetic nervous system. The sympathetic nervous system has the opposite effect on our heart. It causes our heart to increase. And it can actually increase our heart rate up to 250 beats per minute. This becomes, I guess, less effective every year of your life. So I guess the maximum amount of contractions you could have would be 250. And again, that would be like as a little baby fetus or little baby infant. But basically, we take 220 minus your age, and that tells us what your maximal heart rate should be or could be. And again, this is, slowly decreases over time with age. But again, sympathetic nervous system not only increases heart rate, it can also increase the contractile force of the ventricles. So it can also increase systolic blood pressure. And sympathetic nervous system is going to be the dominant system during exercise or during a stressful situation. And again, we know this because sympathetic nervous system's all-encompassing effect on the body is flight or fight. Lastly, the endocrine system can also have an effect on our heart, and the adrenal medulla will release epinephrine and norepinephrine, and these hormones are called catecholamines, and they have the same effect as the sympathetic nervous system does. Again, as you can imagine, it's much quicker for us to activate the sympathetic nervous system in comparison to the endocrine system, but both of them will have a similar effect, and both of them will cause an increase in heart rate and also an increase in stroke volume. Okay, let's talk about blood flow. Okay, so in terms of blood functions and composition, the three functions of our blood include transportation, temperature regulation, and acid-base balance. Okay, so blood is composed of plasma and formed elements. Plasma is basically the liquid portion, and formed elements include blood vessels blood cells and also platelets. Blood viscosity, again, we've already talked about this when we talked about resistance, but blood viscosity is basically just the thickness of the blood. The higher the viscosity, the higher the thickness. Okay. So the body has a tremendous capacity to redistribute blood from areas of low need to areas of an increased need. And this is really exciting and really awesome, I think, because we get some control in terms of where we would like to send our blood. Where are we going to be doing the most work? So skeletal muscle usually receives about 15% of our total blood flow during rest, but this can increase to up to 80% during exercise. And there are two mechanisms of control that would help us to determine how much blood flow we are going to send to these various areas. Okay, so we have intrinsic factors. So of our two mechanisms of control, intrinsic factors are local factors that can cause local blood vessels to vasodilate or vasoconstrict. And then we also have extrinsic factors, such as a neural mechanism that's controlled from outside of the active site. So if you think about this in terms of like a corporation, our extrinsic factors are like the CEO that's like sitting in some office in New York City that you've never met. They can tell us what to do. Or the intrinsic factors are like the local changes we can make within our own offices to get things changed, basically. Aha, this is a nice image that just shows you the difference in blood flow during output or during exercise. So again, you can see the huge change in blood flow to our muscle tissue during exercise. And this is so important for us in terms of our ability to do more work, basically, and deliver more oxygen and nutrients during exercise. Okay, so let's talk about some of these intrinsic controls. So again, these are the changes that we can make at the local tissue. So we can make these changes at our local corporate branch, basically. All right, metabolic regulation is number one. This serves as our strongest stimuli. So if we have a change in chemical concentration locally, this would allow us to increase the dilation or increase the radius of the tube leading to that area. All right, so oxygen is our greatest stimuli. So if you use more oxygen, this can easily stimulate the area to allow more blood flow. Also, other things that can stimulate an increase in vasodilation would be changes in pH, lactate, hydrogen, CO2, and temperature. So all of those would allow us to increase vasodilation to that particular area. The other intrinsic possibility is nitric oxide. So nitric oxide or endothelium-mediated vasodilation. And basically, nitric oxide is produced in the endothelial layer. It's released and causes vasodilation locally. This can be stimulated during exercise. Nitric oxide is produced and released concurrently with calcium, which we know is essential for muscle contraction. So as the release of calcium occurs, we're also producing nitric oxide, which will allow vasodilation. In terms of extrinsic control, again, we're talking about like the CEO that's sitting up in New York City that we've never met before. The sympathetic nervous system is going to play our largest role. And again, we know that he has a pretty all-encompassing or powerful effect on a lot of systems. He's in charge of a lot of companies, we could say, within the body. So smooth muscle that's located in the arterial walls is going to allow us to regulate blood flow because we can change that diameter of the arterioles. Again, if we relax this smooth muscle, this will cause vasodilation. If we contract this muscle, this will cause vasoconstriction or the decrease in radius. So under normal conditions, our blood vessels are in a state of tonic vasoconstriction. Called vasomotor tone. So at rest in our normal lives, these vessels are slightly constricted. When the sympathetic nervous system is activated, neurotransmitters, particularly norepinephrine and epinephrine, are released, and they're going to bind to, this is so cool, they're going to bind to alpha receptors in the gut, which cause vasoconstriction in the gut, and they bind to alpha receptors at the active skeletal muscle, and this causes passive vasodilation. Isn't that so stinking cool? They bind to the same exact receptor, but the receptors have opposite effects based on where they're located. So in the gut, they bind to alpha receptors, and boom, this causes vasoconstriction. In the skeletal muscle, they bind to alpha receptors, and this causes vasodilation. So I just think that's so awesome. So during a hot environment, this becomes difficult. And again, we'll talk in more detail about this in our environmental unit coming up. But we have what's called cardiovascular drift. And this is a progressive increase in the fraction of cardiac output being directed to the skin to help facilitate heat loss. So if you're exercising in a hot environment, we start to have competition for blood flow. We need to send blood to the skin surface to dissipate heat, and we also need to send blood to the muscle in order for us to be able to continue to work at this high intensity. So now your body becomes compromised because it doesn't know, where should I send the blood? Should I send it to the skin so I can get rid of heat? Or should I send it to the muscle so I can continue to maintain this exercise intensity? So not only do we have a compromise in exercise capacity, we also have a decrease in venous return, and this is due to this idea of cardiovascular drift. Basically, like, we want to send blood everywhere, and we don't have enough blood to go around. So therefore, you're sending all this blood out to the capillaries, and not as much is coming back in the veins because you've opened up all these capillaries that wouldn't normally all be opened. And so I guess kind of the take-home message with this idea of cardiovascular drift is that we have a decrease in exercise capacity, or our ability to, compete in a hot environment is really going to limit us. Okay, a couple slides about our responses to acute stress or acute exercise, basically. So what happens within the cardiovascular system? Okay. First thing that's first, our heart rate. So our heart rate is going to increase linearly with exercise intensity. Okay. This makes sense. This is why we use heart rate monitors in terms of training. I'm not sure if anyone in this class has ever used a heart rate monitor for training, but it's the same thing like if you use an elliptical or something in the weight room. If you put your hands on the little sensors, it'll give you your heart rate, and then sometimes it'll tell you like which zone you're working in. So, you can calculate what exercise intensity would be associated with a particular heart rate because of this factor, because exercise is linearly related. The intensity is linearly related to heart rate. Well, how do we see these changes? What happens within the heart itself to allow us to see these increases in heart rate? Well, from our resting state, which, like we said, our normal resting heart rate might be between 60 to 70, to a heart rate of 100 beats per minute, we're going to remove the parasympathetic tone that we have. So we have that vagal tone that was kind of acting on the heart itself. So we're going to remove that. So as we remove our vagal tone or remove that parasympathetic control, that allows us to increase our heart rate to 100 beats per minute. Our heart wants to intrinsically beat at a rate of 100 beats per minute. So if whatever the exercise intensity is only requires your heart rate to be at 100, then that's all you do. You just remove parasympathetic control. If, in fact, you're working at a higher intensity that would require your heart rate to go above 100, we are going to activate the sympathetic nervous system. So between 100 beats per minute to maximal heart rate, this will increase due to sympathetic control. So sympathetic system is going to have its effects on the SA node, which is our heart's pacemaker. What other factors might affect heart rate? Again, we talked about the endocrine system and how the catecholamines released within the endocrine system could cause us to increase heart rate. So like if you have a chronic level of stress, maybe these two hormones within the body would be activated on a more regular basis. So therefore, people that are, very stressed out, even though they don't necessarily need more oxygen, they don't need their heart rates to be higher, they will be higher because of the hormonal factors. Okay, so what factors affect stroke volume? And again, stroke volume is the strength of each individual contraction of the heart. And there are actually 4 ways in which our stroke volume can change. And we'll talk about that here. Okay, so 4 reasons that stroke volume can increase due to an acute stress. So due to exercise, basically. The Frank-Starling mechanism. So, and if you think about this, it's kind of counterintuitive, because if I increase my heart rate, you really have less time for blood to fill into the ventricle. So, I mean, logically thinking, you would think that the stroke volume would actually decrease during exercise. But in fact, it is going to increase. And again, that's why it's important for us to have these 4 mechanisms that would allow us to elevate our stroke volume during exercise. All right, so Frank-Starling mechanism. An increase in ventricular filling causes more stretch on the ventricle walls. So this changes the length-tension relationship within the skeletal muscle to allow an increased force of contraction in the ventricle. This is caused by an increase in venous return and an increase in movement of the blood throughout the cycle. All right, so basically, like, blood is flowing faster throughout the whole system. So we've picked up the current within the river, basically. And so as blood fills into the ventricle and stretches out the ventricular walls, as a result, this contraction will be stronger than it was otherwise. This is the same idea as, like, whether you just stand still and try and jump up, or if you bend your knees and you come down and then try and explode up. So as you stretch a muscle, you're going to, as a result, be able to produce a more forceful contraction in the end, basically. And that's what Frank-Starling is. So you stretch out the ventricle, and as a result, the contraction comes back stronger. So it's the same principle we apply when we do plyometrics, basically. So you kind of squat down and then you jump up. And again, that's going to allow you to produce significantly more power than if you didn't stretch that muscle beforehand. Okay, afterload induced autoregulation. So afterload is the tension in the aorta that must be overcome in order for blood to be pushed out of the ventricle. So if afterload increases, we must, in response, create a larger contraction to overcome this increase in pressure. All right. So there's more pressure in the aorta, so therefore we need to produce a more forceful contraction in order to get blood out. And that's what afterload-induced autoregulation is. Okay. Treppe phenomenon. So in order for the cardiac muscle to contract, and again, we haven't talked about the details of that in this class, that's a little more advanced, something that you maybe would learn in a cardiophysiology class or something like that. But in order for cardiac muscle to contract, we must have the movement of calcium into the cardiac cell. Again, it's a similar concept as what we've talked about with skeletal muscle. The major difference with cardiac contraction is that calcium comes from outside of the cell, basically. But with the Treppe phenomenon, we're bringing calcium into the cardiac cell, and usually following each contraction, calcium is shuttled back out of the cell. So the normal process would be, okay, calcium comes in, heart contracts, calcium goes out, heart relaxes. But as heart rate increases, there's not enough time for all the calcium to exit the cell. So the inside of the calcium, or inside levels of calcium, are going to increase in a stepwise fashion. And that's the Treppe phenomenon. The more calcium basically that's in the cell, the stronger the contraction will be. And that is related to the interaction of the actin and myosin within the cardiac muscle itself. So Treppe phenomenon basically involves a stepwise increase in calcium because there's not enough time for it to be shuttled out of the cell. So as calcium levels increase, the force of contraction produced by the actin and myosin will also increase. And lastly, the sympathetic nervous system has a direct effect on cardiac muscles, and this causes an increase in contractility of those cardiac cells themselves. Cardiac output, again, will increase linearly based on exercise intensity. So again, heart rate will increase and stroke volume will increase. So cardiac output is going to increase in a linear manner based on the needs of oxygen for the active tissue. What about blood changes during acute exercise? AVO2 difference, again, this tells us the amount of oxygen that's dropped off at the active tissue. So we're looking at the difference in oxygen concentration in the veins versus in the arteries. And this is going to increase significantly. It can increase up to 3 times or 3-fold during exercise. Plasma volume, this is interesting. At the onset of exercise, there's immediate loss of plasma from the blood to the interstitial fluid. And this is caused by high pressure inside the vessels, or also known as hydrostatic pressure. We have a 10 to 15% immediate reduction in plasma volume during prolonged exercise. This is one of the important reasons why we warm up, because we know this is coming, and then we're going to actually make some changes based on this. Then we have a 15 to 20% reduction of plasma volume observed in 1-minute bouts of exhaustive exercise. So plasma volume is going to not only decrease during aerobic exercise, it also decreases during resistance training as well. So during prolonged exercise or in hot environments, sweat also causes additional loss of plasma volume. So fluid shifts from plasma into intravascular spaces, and this is basically where the fluid comes from that makes up our sweat. So intracellular fluid loss is one of the most detrimental to us during exercise. This is one of the biggest, risks with dehydration. And again, that's one thing that's really important for us in terms of, during an exercise for us to make sure we're getting the appropriate amount of fluids. Lastly, hemoconcentration will actually increase during exercise. And again, we're not actually getting more hemoglobin. We're not getting more red blood cells. We're just increasing their concentration because there's less fluid in the blood. So the absolute amount of red blood cells or hemoconcentration stays the same, but it increases in relation to the other components of the blood. So because we're losing fluid, kind of like the percentage actually increases. So again, during exercise, the decrease in plasma volume causes a relative increase in hemoglobin or increased hemoconcentration. And our hematocrit can increase from 40 to 50%. So again, that's the percentage of red blood cells that you have within the blood, and this hematocrit would allow us to carry oxygen.